My będziemy tańczyć najpierw. Zaczniemy tańcem krakowiaka. Możemy?
Nie, Ola, weź.
Ja się chowam za kolumną.
Dobra.
Okej. Jeszcze minuta. Ale a może tam z tyłu tak posiadali, widzisz? Ja ich zachęcę tu.
Dobra, ale to już może minuta? Jesteśmy na streamingu już, tak? Dobrze.
Good afternoon. It is a great pleasure to see you all here in our new aula of the Pomeranian Medical University in Szczecin. This is actually one of the first event after the graduation of the pharmacy where we have the pleasure to meet here together. Science is a very, very wide area, especially medical science. That's why we have another meeting with science today, and we've got a really amazing guest. And about him, Dean Professor Mateusz Kurzawski will tell us some few words. But from here, from this place, I would like to emphasize that meetings with science are really important even for both for students and lecturers, tutors of our university, because it really affects internalization of our university, and it really gives us a chance and opportunity to emphasize, to expand our knowledge, and all of you can know everything from these lectures. So, Professor Kurzawski, the stage is yours.
Okay, I'm not a guest here. It's a great honor to welcome to our university Professor Przemko Tylżanowski, who came from Belgium, from University of Leuven, which is very famous university, and the level of research here is really, really high, as I know now. And Professor Tylżanowski is a specialist on using zebrafish as a model organism in biomedical research. And the applications here are very, very wide. There's a broad range of applications. And I think that it's now time to start using that model also in Pomeranian Medical University it doesn't generate very high costs, and it can provide the answers for very many questions we have. So I'm happy that the students of Pomeranian University are here also. For some of yours, it may be the first time you have the lecture that is not online. Yeah, so and it should be like that in the real university, that some lectures shouldn't be online. So once again, it's a great honor to welcome Professor Przemko Tylżanowski, and the stage is yours.
Thank you very much. I already mentioned that I think it's the first time I give a lecture in a place where I don't really see the audience. The columns are fantastic. I wanted to start with this slide. This was my vacation slide in Portugal. I saw a guy who was fishing, and I thought this would be an appropriate leitmotif for our lecture today. We'll talk about fishing and using zebrafish in biomedical research. I am here, I am here from KU Leuven. I also work at the Medical University in Lublin, and I am a chair of a Polish Zebrafish Society, and I just noticed that unfortunately the slides are squashed. The aspect ratio is not preserved. I don't know if someone can fix it, but I will just go on. It's my first, you see the circles become eggs. So this is my first time in Szczecin, and I must tell you, when I was thinking about a fish and Szczecin and my childhood memories, I could not stop myself from showing you a very well-known sculpture of something that we all remember from the early childhood. Now let's move to the lecture. I think this is the problem that we sort of overlook. When we talk about developing new drugs or new treatments, what we need to do is find that drug, use something what is called preclinical models, preclinical research, before we go to patients. And the failure rate of that preclinical research is amazing. If you see that 92% failure rate, I mean, that's absolutely terrifying, right? And it did not change in 30 years. We have all these new machines. I just seen some new fancy machines here at the PUM, but they don't help. We still have very, very low efficiency. For the last time, I will apologize for these slides being squashed. Let's just deal with it. So when you do biomedical research and you try to develop gain preclinical information, you basically are faced with this kind of solutions. You can use something that is experimentally very, very easy and honestly totally useless in preclinical research, or you have something that is very relevant, but you can't do research on it, right? So what are we left with? This is out, this is out. We are really left with this. The lecture today will focus on one aspect of the model organisms. There is also upcoming concept of NAMs, new alternative methodologies, which use organoids and organs on a chip. Preclinical research is done on the interface of companies and universities and all kinds of things. And it's a really research that in Poland is lagging behind. If you look at this, and the blue is in vitro, the yellow is in vivo, and the green is organoids, and this is a 90 and 95% cutoff, you will see that Poland, together with Greece, Hungary, and so on, is really trailing behind in developing new alternative technologies, so organoids here, and it's not really good in using animals. I will show that later. I want to remind you that there is a EU directive basically forcing us to replace animals. And this is something that somehow in Poland is not noticed, which bites into our innovation, because that's what innovation is about, to move forward, not wait for anyone else. Then it's not an innovation, it's a trailing, right? If you look at arithmetics, look at the bookkeeping. This is a size of the mouse market, and in past 20 years it grew 300%. This is a zebrafish market, it's smaller, but in past 20 years it grew 1,700%. If we flip the coin around, for each dollar gained from developing a new drug, into each dollar you have to invest $12.50. If I would run my house budget that way, I would be divorced long time ago, right? With zebrafish, it's a bit better. It's almost getting 1-to-1, and I think we'll cross the 1-to-1 border soon. So if you look at the experimental landscape, and you look at mice, zebrafish, and organoids, mice are really getting out of business. They are still somewhat useful in the metabolic diseases, but zebrafish is catching up. In general, zebrafish overtakes all that, and I'm not going to read the slides. Organoids and organs on a chip overtake that. And you may think that I am bullshitting, right? Well, these are drugs that came into the clinic coming from zebrafish. So this is real, it's happening. Zebrafish also, FDA put zebrafish in one basket with NAMs, with new alternative technologies. So zebrafish is now on the wave that we should cherish and develop, and Poland has all the potential, because right now, if you look at the innovation index for preclinical trials, well, the entire former East Bloc is behind, look what happens here. I mean, we are trailing, we are not following. So the innovation, we have a chance now to be innovative and to actually cash in and not cash for someone else. Today I will talk about zebrafish, and I hope you will believe by the end of this talk, you will realize how much fish is in each and every one of you. You are fish, you just have hands and legs, but actually you are a fish. Why do I say that? First of all, we have the evolution. We share the same lineage, the same genes, the same processes. Animals came out on land around, give or take, 400 million years ago. We are derived from those animals evolutionarily. Also developmentally. If you remember this drawing, it used to be in old high school textbooks. I don't know if in the new improved program it's still there. If you look at the hackles, this is a drawing from 19th century. These embryos look pretty much the same. They are not to scale, but if you look at the morphology, they look pretty much the same. During development, okay, you see this will be a fish, this will be an axolotl, but we still don't know what that will be. Eventually, we know this is a human, this is a mouse, this is a chicken, and so on. Now, think what does it mean? Because this is observation. Now, what's the biological interpretation? Initially, during development, you have different ways to start your process of gastrulation, but then you have a phylotypic stage where we all look the same. And then, of course, it's adaptive diversity. Now, what does it mean that we all look the same? It means that the same genes, the same molecular processes, the same morphogenetic movements, all things happen in different species in the same way. What does it mean? It means that I can use a species A to infer what will happen in species B, right? In other words, if you look at this, and when I was a student, I had to memorize this thing. Honestly, that was a part of the biochemistry exam. The only thing I remember from there is the Krebs cycle. All the rest blissfully went away. But this happens in every living cell, no difference. And if I switch off an enzyme here, it will have the same consequences irrespective of species. So this is why the model organisms can be used in biomedical research. When the genome of zebrafish has been sequenced, it came out that about almost 13,000 genes are the same in a fish that on the total number, by the way, of about 22, right? Are absolutely identical. At the protein conservation, we have about 70% identity. 80% of genes associated with human disease are found in the fish. Are we a fish? We are still not convinced. How about this? Fish have a central nervous system, they have a brain, they have a liver, pancreas, gallbladder, intestine. They have all the same organs we do. Okay, you say they look different. Yes, they look different. But they work the same way, they do the same thing, and they come about through the same molecular events, through the same cascade of molecular events. Which has a consequence later on, because if you look in the drug discovery business, the drug targets conservation between between zebrafish and human is very high. Drug metabolism, which is very important, because if you take drugs, they metabolize, and sometimes metabolites are toxic. Using rats for that is useless, because rats, through the evolution, live on eating toxic things. Mice also don't represent very well human physiology. Fish represent very well. There is also another aspect that we tend to forget. Mice are night animals, which means they are active in the night, they sleep in the day. PhD students are typically daily animals. So they usually work in the day and they sleep in the night. So now imagine someone comes in the middle of the night and is doing experiments on you. I mean, it makes no sense, right? Yes, I know you can have a regulated light in a fish facility where a day becomes night and night becomes a day. I haven't seen one like that in real life. So, no, in reality, the mice work at night. Now, where is the fish actually coming from? What's the history of the fish? Fish comes from Southeast Asia and is swimming in this terribly dirty water, as you can imagine. That's how the fish looks like. This is a female, this is a male. If you look very carefully, you will notice the difference. She is slightly pinkish and a bit more round. The guy is more what we call a torpedo shape. Now, I am so happy talking about a fish, but actually fish came in the science a long time ago. This is the oldest PDF that I managed to find from 1951, where they looked, then it was called Brachydariorerio, then it changed name to Daniorerio, where they looked for toxicity of urethane. Then nothing happened for a long time. Then in the mid-80s, Charles Kimmel came up with a staging table, so we know we have names, you know, 1 cell, 2 cell, gastrulation. Then came George Schlesinger, who did something remarkable that nobody noticed. He actually cloned the fish. Cloning of animal, nobody cared. Can you imagine today? Then he just cloned it. He could show that you can clone the individuals, you can clone the mutations, and so on. But again, it was a very low traction event until that event. Notice the date, 1996. I will come back to that. There is a journal in developmental biology called Development, and there was an issue where this lady and her team, plenty of different postdocs, published the results of a saturation mutagenesis screen. What does it mean? We knock out every bloody gene in the genome, and then we look at the phenotype and we try to establish which molecular and physiological function this gene might have. This was such an important discovery that she got a Nobel Prize for that. And then, kind of interest move on. Now, why do I say 1996? Because today we have 2026. 30 years later, right? Do you know Paul Safo's 30-year adoption rule? It applies to everything: internet, cell phones, electrical cars, whatever. First decade is the wow phase. Oh, cool. Second decade is, well, can I actually make it work for me? Is it somehow useful? Can I make it more user-friendly, and so on? And the third decade, widespread adoption. And this is where we are now in zebrafish. 30 years later, there is a widespread adoption in the world of using zebrafish and zebrafish embryos to do biomedical research. And the rest of the lecture will be on why. From zero to fish, it's about 25 hours, 24 hours. The only thing the fish is not having at this stage is the liver. The liver comes up about 2 days after fertilization. After 5 days, the fish has bone, cartilage, has everything. The heart starts to beat around here, 10, 12 hours post-fertilization. The fish, this is a very old movie, older than most of you in the audience here, but always shown in the zebrafish meetings, because this is the first 18 hours in the life of zebrafish. There is a stage of cell division, epiboly, shield formation, and then here you will have, in a moment, you will see formation of the eye. If you look very carefully, you will notice here, beating heart, somatogenesis, and so on. Why do I show this movie? Notice something very important. It happens outside of the body. It's transparent. What does it mean it's transparent? It means you can do really fancy things. None of the pictures I will show you today, it's either AI generated, computer simulated, otherwise made up. It's all real. For instance, this is very real. This is the same process you've seen before, but it's done by a light sheet microscopy, where we have a fish where every individual cell is labeled, and we can watch it fluorescently, and you can see how it develops, you can see how the cells move, and so on. Because fish is transparent, we can look through it, and I will show you some other examples of that. The other very important thing is that until 5 days of development, you do not need ethical committee permission. You can do everything on the fish. Fish is an equivalent of an adult human being without ethical committee. And this is actually, this should force us to abandon the mice. I mean, you should see the file I had to prepare last week. And then, of course, you can look at fish differently. That was a movie in the light sheet microscopy. You can also do something called single-cell RNA sequencing, where you disperse fish into single cells. You put it in the machine, you push a button, because that's how the technology looks like today. And you get these beautiful diagrams, which don't, it's called UMAP plot. And what happens here is, what you do is with single-cell RNA seq, you sequence all the RNAs from every single individual cell, and then you do so-called unsupervised clustering, meaning all the cells that are similar or identical, you put in one cluster, and then you see what they represent. And that way you can say this is an optic cup, this is an epidermal cell, and so on. So you can molecularly dissect the embryo. I know it doesn't look like the fish, but molecularly I know what's going on. And then if you collect this data together, what you can do is kind of a three-dimensional UMAP plots, which in function of development, you will see changing colors, new cell types will appear, and eventually you can map the organs. This is a periderm, for instance, paraxial mesoderm, notochord, and so on. Why is it nice? It's not only basic science. Imagine I have a mutant where I have an issue with paraxial mesoderm. This paraxial mesoderm, which was somewhere here, will be gone. So I have a molecular readout for my mutation. And then you can put it all together. You can see where the cells come from and where do they go. Again, this is real data. These cells, this is the line of differentiation. And you can watch what happens to the cell. And one of the latest tricks up our sleeve is spatial transcriptomics. What it is, you have a histological section, you do single-cell RNA seq on each and every section, you do this kind of things, you merge it together with single-cell data, and for instance, you can study heart development, and you can see what happens when you regenerate the heart. Heart regeneration, for instance, after heart attack, right? So these are the things you can do at a molecular level in terms of gene expression, present molecules, active signaling cascades. But obviously there is much more that you can do. Let us start with watching the fish. As I said, fish is transparent. There are lots of fluorophores. These fish, RGB, red, green, blue, are just like your cell phones or screens of your computers. And when you have three colors, plus you add some molecular biology wizardry based on the Krylov system, I will not go into details here. What it means is I can mix and match different colors in the cell. And if I mix and match different colors by recombination, I get a color cube. It doesn't look like an Adobe color cube, which has millions of colors, but it's not bad. And if I make a fish using this strategy, I can really label individual cells, I can do lineage tracing, I can see what happens if I have a mutation, if I have a disease, if I have a human disease in a fish, I can see what goes wrong at the morphological level, at the histological level, and so on. Of course, there is traditional way to look at a fish, which is a histological section, but fish implies fluorescence. So the same image, this is a multiple transgenic line where different cells are colored in different way. Incidentally, I show you this eye because fish eye and human eye is much more similar to each other than a mouse eye to a human eye. What happens when you look at the different layers of a human eye and a fish eye, human and zebrafish have the components that mice do not have. Why mice do not have? Because I already told you, nocturnal animals. They don't need the kind of eyes we do. They need completely different kind of eyes. Fish is a diurnal animal, so it requires the same. You see how evolutionarily this is conserved. And then, of course, science is art. You can even win prizes. This is a mugshot. These are eyes, mouth, nostrils, and so on. Of course, you can visualize many other structures. You can visualize blood vessels. You can visualize how nerve tissue goes around, axons, and so on. You can visualize connections, optic tectum, whatever you want, actually, right? You can even make a complex labeling, which is here is a transgenic labeling. This is all live fish, okay? Live, transparent. Where you can label eyes, you can label different parts of the brain, and so on. I am interested in skeleton, so of course I would like to label my skeleton, and here you can see it. Another way to visualize fish is doing nano-CT. This is what I did myself in Leuven. Nano-CT is like a micro-CT, but the resolution is different. Nano-CT has a 1 micron resolution, so it's 5 times better than a histological section, which is typically 5 micron. And then I can dive into this fish, and I can see single cells. As I said, I'm interested in skeleton. You can see the palate here. You can see the cartilage linked to the palate. Another fancy way, and this is what we have in Lublin, it's a confocal Raman spectroscopy. Now, I'm not going to make a fool out of myself. I'm not going to explain to you Raman spectroscopy because, honestly, I don't understand it myself very well. But the imaging you can do here is more focused on types of tissues. So what you can do is you can look at proteins, lipids, you can look at collagens, you can look more globally, which is very important if you study, again, consequences of the mutation. So this was the imaging. Now let's move back to the biomedical research and how useful can be fish in biomedical research. Let's look at the heart first. Yes, fish have a heart. Some professors don't, but all fish do. And this has an atrium and a ventricle. And I said because it's transparent and because it's live, you can visualize it in many different ways. You can see it beating. You can see the valves here, how they move. You see, open, close. The microscopy went so far away these days. We have so fantastic imaging technologies and strategies that you can really see this in real time while the fish is, for instance, sick. What can happen, what can be wrong with a fish heart? Well, fish can get a heart attack, can get the inflammation of the heart muscle, can have a scarring tissue, pretty much the same as humans. If we block one of the calcium channels, what you see here is the red blood cells. This is a normal situation where the blood is very nicely expelled from the chamber. Here is not expelled because we block one of the calcium channels. So this is the kind of things you can imagine. Of course, you say, yes, heart, I typically associate with electrocardiogram, sure enough. This is a very high-tech setup. I don't know if there is a cardiologist in the room. Probably can read these things, I can't. But again, you can do electrocardiogram. You can give drugs to the fish. You can see how it affects the readout. And it's very likely, and in fact we know in most cases, it will have the same effect on humans. This is work from Lublin here by Monika Maciąg. And you see she was giving a drug called doxorubicin, which slows down the heartbeat, and this is a beating heart. She would, read it out, and she would show that the doxorubicin decreases the heartbeat. So again, you can imagine you have a readout, you have a setup to look for drugs modulating the heart rate. The heart is, in terms of organization, physiological organization, the same as in human. Just to stay for a moment with the broadly understood cardiovascular system, fish can suffer from another condition, and I think last year or 2 years ago, there was a year of the stroke. You know what's a stroke, right? A massive hemorrhage in the brain. So fish can get a stroke. And when a fish gets a stroke, it's massive. You see the blood accumulating in the brain. This is a transgenic fish. It's a GATA1 labeled with DS red. These are blood vessels. You see the blood is all over the brain. But you can prevent it. And you can prevent it by giving mycosonol, and it's gone. And this is in clinical trials now with people from so-called high risk group for stroke. That's how far the conservation is in physiology and biochemistry between these two species. What about metabolic diseases? For instance, diabetes. When you look here, you have the hypoglycemic, so you have a high glucose, and you see you get what patients get, which is a retinopathy. And then you get alifiricept, which is a drug that you use in the clinic with humans, and you start to slowly cure that condition. There is also a model for insulin deficiency. There are several other models addressing the neuropathies in zebrafish. Fish can also get fat. We call it American phenotype. Look at the liver. It's on a high-fat diet. It's a very fatty liver. This is an oil red staining, so fat is red. This is normal fish, yes, there is some fat. Look at the fish on a high-fat diet. It's red all over the place. When you get obese, when fish gets obese, number of behavioral changes are similar to those in people. Here you have a readout of a movement of a fish in a fish tank. You can see the outline of a fish tank here. The red is where fish stays a lot in one place. The dark blue is where she moves around, or he. So you can see that the fish in general more or less keeps moving around the entire tank. If the fish is on a high-fat diet, however, it's on the bottom of the tank, and it stays there. For people studying fish behavior, that means fish is very, very anxious, nervous, goes to the bottom, tries to hide. If you put fish on a high-carbon diet, a high-carbohydrate diet, fish does exactly the same with children when you give them too many candies. Goes to the top and runs around trying to expel all the energy it has. So again, there is a big conservation between a human and fish behavior, and really you are, a lot of you, I mean, a large part of you is a fish. While preparing the lecture, I discovered something I didn't know it exists. It's called traumatic acid. I thought it was some spelling error. No, it's not. Traumatic acid exists. It's a natural compound that is used in wound healing. But one of the recent discoveries shows that it's maybe a competitor to Ozempic, because look what happens. This is HFD, it's a high-fat diet, and if you give a traumatic acid, the dark red here drops down. In other words, these are some control fish, but look at this too. This is a diet-induced obesity, and you see this red here. You add traumatic acid, the fat is gone. And because it's a natural compound, it's sort of easier to bring it to the market, not easy, but easier. So who knows, it might be a next competitor to GLP-1 or Ozempic. Another very nice example how you can go from fish to a patient is here. You have this lymphatic anomaly. I don't know if there are doctors in the room, but if there are, they probably recognize it. This is a patient, human patient, with massive issues in lymphatic system. And that's how the patient looked in real life. What has been done is there is a lymphatic model in zebrafish, and this model was used to test various pharmacological solutions to help the guy. So you see here, for instance, it's mess, it's all green, it doesn't look good. The moment number of drugs was tested, one of them brings back more or less a normal situation. From the fish, this drug was given directly back to the patient, and this is what happened. So, and this took 2 weeks, even not. So that's how quickly you can move from the fish to the clinic and to the patient. And honestly, what patient cares about can walk. It's almost like a miracle, right? Now, what about another big field in zebrafish, which is neurology and neurobiology? Fish can think, and we can see it. Sometimes I wish I could see with students that they think. Unfortunately, their skills are not transparent, but were they transparent, I could see something like this. This is a live image. This is a calcium channel reporter, and you see when fish is exposed to a signal, suddenly the brain flares up like a Christmas tree. And this is olfactory. So the fish is smelling something, suddenly it smells something more, and there is a flash. So you can watch it in real time, live, how the neurons fire. And again, if you are in any way associated with biomedical research, you think brain, you say, well, can you do electroencephalogram on a fish? Yes, you can. This is a setup we have in Lublin. There are 3 like that in Europe. And you can measure, you can do electroencephalogram, and you don't do it just for fun. There is a big epileptic research, because there are many patients suffering from so-called drug-resistant epilepsy. And there are now 2 drugs on the market that came out of that research on zebrafish that actually help the patient. So this is how you see it. This is our electroencephalogram. This is the calcium reporter that you've seen already. And this is a seizure, epileptic seizure. So you can induce epileptic seizure in the fish. And when you induce epileptic seizure in the fish, you get this spike and you get this flare. So of course you can look for a drug solution, right? In general, fish can suffer from other issues. They can suffer from ADHD, aggression. Fish can get very aggressive. PTSD. Aggression in the fish is very simple. You put a mirror, and like some of us in the morning, when they look in the mirror, they get this wave of aggression. Fish is doing exactly the same, very, very aggressive, tries to bite itself. The moment you remove the mirror, nothing happens. Now, whenever you see something, think, how can I use it? If I have a setup like that, I can use to look for drugs that modulate aggression, right? Very, very important. The same is with drug addiction. In Lublin, we have a program on cocaine and mephedrone addiction using zebrafish. Fish get very addicted to drugs, just like humans, and they have the same withdrawal syndromes. So you can look for drugs that would modulate this withdrawal period. And fish can become alcoholics, no problem. And if you give them too much alcohol, they behave like people, which means they stop moving eventually. So it's really very, very, physiological response. Humans and fish is really, really pretty much the same. There is another intriguing experiment that was done, and that's a little bit in the light of how should we, anesthetize the fish. And this experiment was done using something called a Campari. Some of you may remember what Campari was, the older generation anyway. The younger one is the Aperol generation, I guess, right? It's the same thing. So you have a Campari, and what happens is it's the same kind of a fish reporter, and when the fish is anesthetized, it's all green, nothing happens. The moment fish is happy, freely swimming, you start seeing some pink things. Pink things means the brain works. You receive some impulses, there is some calcium movement. The moment you hit the fish, it starts to get really much more pink, especially in the hind and midbrain. Olfactory sort of stops working and so on. If you induce a seizure, epileptic seizure, the entire brain lights up. That's not surprising, right? It's just all pink. What is very surprising is when you cool down the fish, which is a recommended method. I am looking at the head of the fish of the animal house, which is a recommended method for anesthetizing the fish. When you cool down the fish gets a seizure. So is that the best way to anesthetize? I am not sure. But anyway, this is a nice readout, again in a live fish that you can watch. What about infections? We know the word infection invokes kind of a PTSD-like behaviors because we are all after COVID, and we still don't feel very comfortable with this. But we can use zebrafish to study that. Not actually COVID. The best animal for COVID is hamster. But fish have an innate immune system. I told you day 5 is the magic day. Until here, you don't need the ethical committee. Well, development goes pretty much like in humans, right? Fish has nice macrophages, which you can color, of course, and watch them, how they go around and do things or do nothing because there is no any kind of a danger. Okay, now it's moving. You can watch them, no infection, they just... There was a very intriguing discovery related to immune response made just 2 years ago. Acid comes out, Come on, move forward. Okay. As it comes out, there is somewhere here Nilo. Fish have tonsils, just like humans. And what's most amazing, these tonsils have the same function, which is preventing infection. That's how the tonsils look like. These are these purple things. Thymus is here. You have your tonsils here. The same function. Would you think fish has a tonsil? I wouldn't, honestly. And so I'm very happy it got discovered. Because we have all the components of the immune system, at least the innate response, we can study all kinds of infections. This is, for instance, meningitis. Yes, fish can get meningitis. This is a pneumococcal infection. Green is the bacteria which invades the entire brain. You remove a receptor binding, you make a mutation in receptor binding protein of the pneumococcus, and it cannot enter the brain anymore. So again, a very nice model system to look for meningitis, as it is a very nice model system to study sepsis. This fish undergoes sepsis. It's all green because fish is a ROS reporter, reactive oxygen species reporter. When there is a high level of reactive oxygen species, fish gets green. So once you have the system, you can play, and you can look for a drug discovery where you look for drugs that will eventually block the sepsis. And once you block the sepsis, you have the drug. You know it doesn't do any other damage to the fish. You can easier move to the humans. In fact, brain infections, lung infections, urinary tract infections, all this you can do in the fish. And these are the examples of, for instance, here you have a salmonella infection, and you have neutrophils that come to action. So the same responses, the same kind of signal response in zebrafish as in humans. And last but not least, cancer. Now you would think, how can you use fish to study human cancer? There are actually many ways. In fact, I don't know if you know, there are quite nice work done on Drosophila, on fruit fly, on human cancer. But today we'll talk about fish. And when you think about cancer, I would like that you realize something. Imagine this calendar, and imagine in January, you get a cancer diagnosis, initial assessment. I talk about standard care, standard level of care. I don't talk about a Polish cancer kind of oncological system. But in general, you get the initial assessment, you get a treatment selection, so you decide what to do with the patient, and then you begin the treatment. And I think we all agree here. Then are adjustments. Why there are adjustments? What does it mean? Because many patients suffer from side effects. 15 to 20% suffer from side effects. Let it be metabolite, because the drug is metabolized in patient liver and the patient doesn't deal well with metabolite. Genetic composition of the patient. In any case, 15 to 20% need to stop because the drug is toxic. And then we go into optimization. Up to half of the people needs to change it then, because by then it comes out that it, although initially looked okay, it actually doesn't work. January, July. For a cancer patient, that's eternity. We cannot wait, right? And I talk about, let's say, standard of care, 6 months. That's what happens. Can we use zebrafish? Well, you know melanoma. 50% of melanomas are caused by a point mutation in a BRAF6 gene. So one point mutation makes the cancer. Take a human gene, put in zebrafish. This is what happens. Fish gets melanoma. Now, I would like to pause here for a moment. What does it mean that a point mutation causes a cancer? Point mutation per se doesn't do anything. But the point mutation, the gene product, It's a part of a network of interacting proteins, signaling cascades, and so on. One point mutation screws up this network completely. In order for zebrafish to get a melanoma, the zebrafish must have the same network as humans do. Otherwise, the point mutation would do nothing, right? So it has to be the same. Otherwise, it wouldn't work. And here is probably the most intriguing experiment. This is the primary melanoma. Now look here. What kills a cancer patient? Have you ever thought about it? It's actually very rarely that a cancer kills a cancer patient. Cancer has to be located in the brain or something. It's rare, very rare that a cancer kills the patient. What kills the patient is metastasis. So the cancer cells go somewhere else and screw up entire body. And the second thing that kills cancer patient is chemotherapy. It's toxic. It's very, very toxic. So you have a combination, you have the metastasis and you have a chemotherapy, which are the lead killers in oncology, right? Now, this metastasis is very important because in mouse, you cannot really study metastasis. It's a very complicated way to study metastasis in mouse. When you do cancer research using mice, you can do proliferation, vascularization, all these kinds of things. You cannot do metastasis, and that's what kills the patient. In the zebrafish, human cancer metastasizes. So for instance, I take a patient's cells, I color them red, or whatever color you want, and I inject in zebrafish. And I inject, and they start moving, and they metastasize. Okay, they go into blood vessels, they get out of the blood vessels, they colonize tissue. In other words, I have a fantastic setup to block metastasis. And this is actually a procedure which is implemented in several countries already in Europe. It's a clinical procedure where you take cells from a patient, do a so-called xenotransplantation, do drug testing in the patient, and you develop a personalized treatment. Personalized medicine is something very hot, you know. You develop a personalized treatment, and you help the patient. Does it work? Well, this is work of Rita Fior. She started with breast cancer. Now it goes to other cancers. These are two different patients with injected tumor, and these are different drug regimes. Look what happens. This patient doesn't respond to that drug. This patient does not. Here, this combination works on both. Which means, on this patient, I don't have to try for 3 months, does it work or not? She may be dead by then. All this, from the beginning to the end, takes 2 weeks. In 2 weeks, I know what to give the patient, and in what dose, and in what ratio, and so on. This is implemented in Portugal, in Spain, France, Belgium, Holland. Poznań is starting doing that right now. So pre-testing drugs on zebrafish before going to patients. You can do it by hand. Of course, you can have machines to do a really high throughput screening. You put the embryos in, machine injects. You don't even have to inject. Machine injects, and you just look for the best drug combination. So when we come back to that scheme, with zebrafish, this is gone, this is gone, this is gone, this is gone. What we have instead, begin treatment. AV stands for avatar. 10 days to 2 weeks, and you begin treatment. February. For cancer patient, that's a day and night difference, believe me. And that brings us to a general drug screen, because fish are sort of most commonly used in a drug screen, where this is your typical, cost effectiveness of various stages. In a fish, you really cut it out. And what's very important, FDA now pushing to skip rodents altogether. And a new directive from EU, after 2030, it will be very, very difficult to use mice for any biomedical research, just because there is no point. It's just too expensive and too inefficient. So that will go away. So you have these all kinds of things. When you think about drug screening in the fish, remember there are different readouts. You can look at morphology, but you can have a reporter. It can glow green or red or stop glowing. You can have a phenotype that you rescue or you induce. Possibilities are actually endless. and you can do all kinds of metabolomics, and you have it. Again, this is done by machines. You can be done by machines. It's done by humans. It can be done by machines, where you pick up the fish, put it in a capillary, rotate any way you want, put it under the microscope. That's how it looks like. And then what you do, and then use AI, because it works very well, to quickly identify fish that have a phenotype. AI will tell you what potential mutation that is. So for instance, you can screen using zebrafish. It's a 96 well-plate format. You put it on Friday. You can screen basically, 10,000 compounds a week, okay? So you can really quickly look for drugs. And to really close it and to justify the reason why I give a zebrafish talk, I will show you 2 slides on my work using zebrafish, convincing you that a fish is human, or humans are fish, either way you want. We have this family with clubfoot. Clubfoot is a condition where a baby is born and the feet are bent inwards. We discovered a family, did a whole genome sequencing, found a mutation, and when we put this mutation in the fish, we damage the muscles. When we put this mutation in the fish, the integrity of the cells, this is a special dye that should stay in the cell when the muscle is intact. In the mutants, the dye is all over the place. We did myograms in patients. We did myograms in the fish. They have the same parameters. So by patients, simply when the mutation causes muscle to be more stiff, less functional, that's why the foot is bent. And another example is of a cleft lip palate. So you have a cleft lip here. And what we have is we have a mutation and patients with a cleft lip. And when you introduce this mutation in the fish gets a cleft lip, exactly like in a patient. I mean, I don't know how much more convincing you need to see the, if not similarity, even identities between humans and fish. And just to, as a cherry on a cake, fish becomes so popular that actually flew on a space station, of course Chinese, space station, and this is fish in space. The bubbles are round because things in space are round. And you can see the floating fish. So the whole idea is that, you see the fish gains so much traction that it will go into the space. What about zebrafish in Poland? Because you may think, I am just coming here saying things, and in fact no one else is doing that, right? Well, in Poland, this is the number of publications that come out. This is a post-COVID consequence here, that come out in Poland. This is Lublin alone, so about up to a half of publications in Poland are generated in Lublin when we have the facility. But there are some fantastic fish facilities in Poland. Piotr Podlasz in Olsztyn, Tomek Preisnar, Magda Chadzińska, Krzysiek Rakus in Jagiellonian University, International University in Warsaw, several labs, Cecilia Winata, for instance, Jacek Kuźnicki. Fantastic research, and I really urge you to, because this corner of Poland is kind of fishless. You have a fishless phenotype, and it will be time to fix it. And as I said, there is no Photoshop on this presentation. This is not a Photoshop. This is whole mount in situ hybridization on a zebrafish embryo. This is a FOXJ1 gene. This is a precordial plate. These are the somites. And when my PhD student was finishing her PhD, and she saw this, she came to me, she said, look, even fish smiles and mean I can finish now. I said, yes, you can. And with that, thank you very much, and on time. And if you have any questions, I'll be happy to answer them in any language, either Polish or English, okay? Thank you very much.
Is it? Yeah. Dear Professor, thank you very much for extremely interesting lecture. I must admit that you really inspired me, And my main aim of my interest are immunosuppressive drugs and generally transplantation. So I started to think, how can I develop this area of the medicine in our university? So thank you once again. It was really, really inspiring and amazing lecture.
Thank you very much. Yes, you can. You have a fish facility at your university. I've seen it today. So you just have to start using it.
Exactly. So we've got some person who is responsible for this area in our university, and hopefully we are waiting, and we can't wait actually to make it happen.
Students, any questions? Don't be afraid. I don't bite. I'm vaccinated.
Students or tutors? We've got Dean.
No, come on, guys. No questions?
May I begin?
Don't be afraid.
The last time I was a student was 30 years ago. Coincidence? I graduated in 1996. Thank you for a fantastic lecture. Now, I have two questions. One of them, because I teach bioethics here, and from time to time I also teach bioethics to biotechnology students, and we are discussing the ethics of animal research and why we have this trend to eliminate animal research altogether. And one of them is that, the bioethical philosophical foundation of this EU directive that you mentioned was that we know more and more how animals, vertebrate animals, are similar to humans, to the point that they actually meet some very important personhood criteria in philosophical sense. That means they perceive the world the way sort of we do, they can feel pain, they can suffer, and this is why we should avoid harming them, especially if we harm them for our benefit. So now, what is the loophole that makes zebrafish, on one hand, like human, but not human enough for us to ask the bioethics committee? What makes the difference between zebrafish age 4 days and zebrafish age 6 days?
Very good question. Actually, the definition is self-feeding. So the moment the fish can eat itself, it becomes sort of protected. Okay. I don't know if you know, since you teach ethics, that chicken embryos are not considered animals until hatching. Okay? So we are suffering from a Bambi syndrome. For the students who don't know what a Bambi syndrome is, but I'm sure you all know what a Bambi is. Bambi is something cute. So if something is cute, we cannot harm it, and if something is ugly, like some snake or something, then we don't care. Which is, of course, hypocritical, but that's what it is. Now, until the definition is self-feeding, in Canada is, I think, 6 days. Maybe in Canada they start eating later. But in general is this rule. Now, there were attempts to reduce the time allowed for first 3 days, but thankfully it died. If this would be implemented, the entire drug research would die, right? Because it's just, you can't. So, ethically it's always very complicated, and ethically you can always look at the same thing from 5 different angles, and all depends how you feel given day. Do fish sense? Well, you've seen the brain. It flares up. If you play a music, the brain will resonate together with music. I think Piotr showed last time a slide like that. Is the fish conscious? I don't know. Is Claude conscious? I don't know. Or ChatGPT? I don't know. We are entering a very... Students these days, I think, the ocean of uncertainty and mixed decisions and messages is much bigger than during our times. During our times, it was more blackish, whitish. And now the colors are gone. So as long as we can do it, we'll do it. Of course, it would be silly argument, I don't care, fish doesn't feel. I don't know. if I told my car navigation, because I try to get somewhere and voice command, and I finally said what I think about her, then she said, please contact program developers. So,
at the risk of monopolizing the question and answer, I have one more. You told us about like bazillion of applications stemming from the fact that these fish are so like humans. 80% of human disease. So my question: what are zebrafish not good for in human
very good
disease?
question. So, in terms of cancer, if I take patient biopsy and put in the fish, no limitations. But fish don't really have lungs. There is a swim bladder, which has an external vascularization. By some is considered a very primitive lung, but it's not really a lung. Fish do not have prostate. What is fish not good for? Fish is also, in terms of behavioral studies, after several decades of work, I think we pretty well can, for instance, fish can get autistic. We can have autism in fish. One of the definitions of autism is lack of causative memory. What does it mean? You have an autistic child, comes to the kitchen, touches a hot pot, screams, comes next day, touches the same pot, screams. No cause-effect connection. It's not learning. There is a setup in the fish, speaking of ethics, there is a setup in the fish where you have a dark chamber and a light chamber, and the dark chamber has an electrical current. So when the fish swims into a dark chamber, gets a shock. And a normal fish swim maybe twice in, and that's it, and they will never go to dark chamber. The autistic fish keep on going into this dark chamber, they don't make a link. And then, of course, you have a model to develop the drugs. But what is fish not good for? Late immune responses, because, well, you can use it, but then you need ethical committee, right? Fish, the kidney works a little bit differently. Some aspects are the same, some are different. Liver metabolism is to a large extent the same. Eyesight is the same. I think, the same mutation in the fish not always will cause the same phenotype. In my research, I got very lucky. I get the same phenotype. But there are cases where you get a different phenotype. You can't predict it. The reason is that, let's say the biggest disadvantage of fish is that it has a partially duplicated genome. What it means is during evolution, the genome for one or another reason got duplicated. Now, what does it mean, partial duplication? So some genes exist in two independent copies, which inherit independently, and they form an A and a B variant of the same thing. And the example that is very familiar to me is noggin. It's a gene, it's an antagonist of BMP. Humans, mice have a one noggin. Fish have three noggins. They all do the same thing. They are just expressed in different places through different genes. So partial duplication complicates the issue. Pattern of splicing is often different. So yes, there are limitations, but then I think because it's fast and cheap, it's very easy to see if in your particular research, if for your particular question, is fish useful or not? And I would say within a month you have the answer. If it is not useful, of course you don't do that, right? But it's about, so the speed I think is the key advantage. Students? Yes. You should get an award for the bravest student in the room.
Thank you very much for the great lecture. I'm myself quite interested in epigenetics, and I was wondering whether fish possesses the same epigenetic landscape as humans, so DNA methylation, which basically silences genes. Are there ways to study, for example, how environmental factors affect epigenetics in fish?
It's exactly the same. Of course, the methylation pattern may be different, also because of that partial duplication. The chromatin structure, it's also conserved, as you know, across cell types or species. Number of the large chromatin structures are pretty much preserved. Methylation epigenetics, very much so. Different types of RNAs, long non-coding and so on, are present in the fish as they are present in the humans. All these things, evolution is very lazy. Once it invents something, it stays there. All these things were invented long time ago, and they come in the fish and they come in other species. Number of methylations you can do even in Drosophila. So it's very conserved. Yes, you can do that.
Thank you. I've already seen another student. I must admit that this is the first lecture during when nobody left, actually. So Thank you very much.
Maybe because there is a paper they have to sign.
Okay, this may be a very stupid question, but I was curious. It was stated already that drugs that affect the lungs are not the best because, of course, fish don't have lungs. But there are many medications that are not specifically for the respiratory system, but they affect it in some way. And I was curious if you can in some way check the dependency between the other organs and the respiratory system. How does it affect it? If there are any similarities between lungs and gills, I don't remember how it's pronounced.
This is one of those limitations which we very nicely found. The problem is that fish, as you know, breathe differently. And the swim bladder is considered a primitive lung because it has its own vasculature, and there is a limited gas exchange, but you don't have a classical gas exchange like you have in land animals. So the only aspects, for instance, you cannot study the aspects of lung function when it comes to alveoli, when it comes to all the proteins in the lung. The only thing you could study is the metabolic effect of drugs on the other organs, because the drugs are metabolized in pretty much the same way as in the fish, as in the human. So you could test that to see, for instance, if there is a problem with the liver or in the kidney or what happens. But in terms of a real lung function, for that we would have to go to the lecture I didn't give, which is on mini organs, because you can make mini lungs, functioning mini lungs from your own skin cells. Honestly, yeah, we're using the Yamanaka cocktail. You can make your mini lungs, and there you could, your question would be answered in a more relevant way on the lung. But if you talk about metabolism, then you simply give the drug to the fish, and then you look on the other consequences, but you do not have the lung readout.
Thank you very much.
Yeah.
Are there other questions? Dear students, you've got a unique chance to ask the question.
First, thank you for the lecture. I have a question about cancer treatment and how is it different, the alternative splicing or gene realization that affect the treatment of cancer, like, for example, in human genes, like mutation of BRCA1 or BRCA2. Is it any different, or you found something interesting that it's correlating differently for another species, and does it affect like how to say it? How goes the does this correlate with alternative splicing or NSIPs or CNVs?
Okay, the most classical example of cancer-related alternative splicing is FGF receptor, right? I cannot answer the question because I don't have the knowledge on how differential splicing, to what extent the splicing in a fish would be different than in humans. I don't expect a big difference, but I don't have hard data. The key use of fish in cancer research is in the treatment. So we are not studying the molecular mechanism of cancer in the fish because, that's for veterinarians, honestly. We don't do that. We more focus on treatment. So we induce cancer in the way we know happens in humans, like a point mutation in melanoma. Or we can induce some mutations, FGF receptor or any other, RAS or whatever else. And then we focus on treatment. So in that sense, we not necessarily study the mechanism of cancerogenesis. We more study the mechanism of treatment. Can we fix it? What can we do? We look at ROS-reactive species, we look at metastasis, we look at all these things. But the fish is used in that preclinical sense from the other end. So we don't look how it happened. We already have a patient where it happened. We rather try to help the patient not to die. So it's more used that way. If your question is about the mechanisms, I would imagine there are differences. As I said, I am not qualified to answer that question. I would imagine there are differences, but I would be very surprised if there would be a big difference. Yeah.
So, I can't see any other. Yeah, we've got someone.
They are waking up.
So you said that the xenograft, the larva model is great to check if the drug is good for the patient. What are the limitations of this model?
Limitations are, there are a few limitations that you need to remember. One limitation is not all cancers can be tested in that way because the fish, the maximum temperature in which fish can grow and not die, it's about 33 degrees. Let's say in this type of experiments, 33, 34. We are 37. It sounds like a small difference. There was a paper published recently that in fact a 1 degree difference changes the stability of the membrane and makes the tumor benign versus malicious. Okay? So one of the things to remember would be keeping in mind that temperature difference. In case of Rita Fior, the breast cancer clearly works very well in this system, and I think part of her luck was she picked accidentally, obviously, the type of cancer that works in the zebrafish. It could be she would have started with a cancer that doesn't work in zebrafish. Most of the cancers I know work in zebrafish, but I'm sure there are exceptions. A very good exception is a glioma. Glioma doesn't really work in zebrafish. Glioma is not spreading in zebrafish. It grows like a lump. Maybe we do something wrong culturing the embryos, I don't know. But that would be, for instance, cancer that would not be a primary choice. Prostate, lung, that all works very well in the zebrafish. Another thing is a little bit depends which aspect of cancer you focus on. Do you focus on proliferation? Do you focus on metastasis? I would be mostly interested in metastasis as the key killer, but the other factors are also important, right? So you always need some preliminary study. There is no reason to suddenly do only zebrafish. No, we have to adapt as we go. Pick the best system for a given question. Define the question very well, and then pick the best system. Sometimes organoid is enough, right? Sometimes something else. But I think the advantage of zebrafish is when you do the drug screen, you see what happens to the rest of the fish. And so, in other words, you don't kill the patient in the process. So these are the things, to keep in mind. There is no black and white. It's case by case and step by step. It should be one of the options on the table that you test because it's fast, quick, and easy. And it doesn't pan out, don't beat a dead horse, just move to the next one.
So with this sentence, I think we will finish.
Thank you very much.
Once again, thank you very much for coming here, and it was a really great honor to host you in our university.
Thank you so much for the kind invitation. Thank you.